Same Brain, Different Wiring by Richard Jonathan O. Taduran, Ph.D. If you laid 100 human brains on a table, they would look nearly identical: soft folds, mirrored hemispheres, the familiar geography of a species with 300,000 years of shared ancestry. Yet beneath that surface symmetry is a universe of variation. Neural wiring is not a fixed template. It is a biography. It carries the marks of hormones, nutrition, childhood play, social expectations, schooling, stress, work, aging, and the lived ecology of gender and culture. Culture wires the brain as much as biology does. Our languages, rituals, hierarchies, and daily routines repeatedly sculpt neural pathways. At the same time, the brain shapes culture. Our shared stories, norms, and institutions emerge from the cognitive architecture we all carry. It is a feedback loop, each side shaping and reshaping the other across generations. Human brains vary, and they vary bioculturally. Sex differences in the brain. Neurological sex differences exist, but they are neither absolute nor deterministic. They arise from the interplay of biology and culture, not from a rigid split between male brains and female brains, as some self-help books like to claim. Biologically, studies show subtle tendencies in how information flows. Ingelaliker et al. Found that male brains tend to have more connections running within each hemisphere, while female brains tend to show more connections running between hemispheres. These are tendencies, not rules. There is enormous overlap. Zaidi's 2010 review added that men have, on average, larger brain volume, while women have relatively more white matter for their brain size. But these statistical differences do not predict ability, intelligence, or personality. They simply remind us that biology is varied, even within one species, but biology alone is insufficient. Across cultures, boys are encouraged to explore outdoors, take risks, and engage in spatial play. Girls often receive more relational, verbal, or caregiving tasks. These patterned experiences shape neural development through plasticity, gendered nutrition, early adversity, unequal healthcare access, and chronic stress, documented in public health data from WHO and UNICEF, further modify brain development across societies. Reproductive life widens the divergence. Pregnancy, postpartum shifts, and caregiving expectations alter stress physiology for many women. Men, in many societies, face occupational risks and provider roles that bring their own neural consequences. In reality, sex differences in the brain are biocultural footprints produced by hormones interacting with gendered lives. Age differences and the 4 turning points. Age differences in the brain follow the same rule. Biology sets the stage, but culture co-directs the script. Earlier lifespan studies showed that brain networks do not develop or age in a straight line. Cao et al. Showed that some networks become more efficient, while others become more specialized. Baum et al. Found that children's brains gradually separate into clearer functional modules, supporting stronger executive function. Fan et al. Showed that the brain's overall complexity rises and falls in waves, rather than one predictable trajectory. A new study by Muesli et al., published in Nature Communications, strengthens this view by identifying 4 major turning points at around ages 9, 32, 66, and 83, 9 years. Early childhood is dominated by pruning, as the brain removes inefficient connections. Culturally, this coincides with intensified schooling, gendered expectations, and early puberty, 32 years. From late childhood to the early 30s, the brain becomes faster and more efficient. But many societies expect full adulthood by the early 20s, long before this neural refinement is complete. By the early 30s, the brain reaches a steady balance, still capable, but no longer accelerating. Cultural timelines around independence, work, and family formation intersect with this neurological shift. 66 years. Midlife brings slow changes, slightly less integration, slightly more specialization. This aligns with anthropological models of midlife: stable roles, caregiving burdens, career consolidation, and class, and gender-shaped stress exposures. 83 years. Older adulthood brings increased vulnerability in communication hubs. Cultural factors, retirement norms, elder care systems, family support, community inclusion, strongly influence whether this period brings decline, resilience, or both. After 83, individual life histories dominate. No two brains age the same way. Put simply, brain aging is not a steady slide. It shifts in phases, each one shaped by biology on the inside and culture on the outside. The biocultural brain. Human variation in the brain is not an error in the design. It is the design. Sex-linked patterns of connectivity interact with gendered life pathways. Age-linked turning points unfold within culturally constructed timelines of schooling, work, family, and aging. The new evidence for 4 major rewiring points across the lifespan reinforces what biological anthropology has argued for decades. Brains develop within ecologies. Hormones matter, but so do playgrounds. White matter peaks, but so do cultural expectations at 30. Neural vulnerability rises in old age, but so does the protective force of social networks and meaningful roles. For clinicians, educators, and policymakers, the implication is that human brains must be understood bioculturally. There is no average brain, only the shared architecture of a species expressed through diverse, culturally situated lives. And across those lives, the brain turns, reorganizes, and adapts, carrying each of us through the many selves we become.